Thank you very much, Claude, and thank you, all of you, for your time. So let's start with my PhD presentation. As you know, coral reef ecosystems are among the most diverse ecosystems on Earth, with high biodiversity and providing important ecological services. Among coral reef organisms, we must not forget sponges. So if I ask you, do you see sponge in this picture? Yes, here it is. So sponges are sometimes hidden, but they play a very important role in coral reef. Sponges are the most ancient multicellular animal on Earth. They are efficient seawater filter feeder with a simple level of organization. The sponge body is organized in the aquifer system that consists in channels and chambers to allow seawater circulation for feeding and respiration. The seawater enters from the ostia and goes out from the osculum. The sponge skeleton is made by both organic and inorganic materials, such as calcium carbonate and silicate spicules. I will talk about sponges that are made by silicate spicules. Sponges also represent one of the most complex holobionts in coral reef, which means the sponge host plus all the associated microorganisms. So why sponges are important in coral reef? Sponges provide macrohabitat for coral reef organisms. They contribute to the benthic pelagic coupling through the biological processing, such as the silicate, phosphate, nitrate, and carbon cycles. My thesis then, it will focus on the carbon cycles, because sponges are able to transform dissolved organic matter in a more available food source in coral reef. In recent years, two important pathways have been described to explain how sponges recycle carbon in coral reef. The first is the sponge loop pathway, mainly described for encrusting sponges. So here we have the sponge that take up dissolved organic carbon, or DOC, produced by primary processes, transform it and release it as a particular organic carbon, or POC, which is also called detritus when we subtract the live particular organic carbon to the particular organic carbon. In this case, carbon is available on coral reef and to higher trophic levels, and then sponges play a direct trophic link between corals and detritivorous feeders. But not all sponges do the sponge loop pathway. An alternative sponge loop pathway has been recently described in massive sponges. Here, sponges always take up this DOC released by primary producers, but in this case, it's not released as a POC, but carbon is mainly stored in the sponge biomass. So in this case, the transfer of carbon to higher trophic levels occurs through spongivores or at the death of the sponge. So here, we have less POC in the environment, and this could have a cascading effect on the organisms that feed on it. So these two pathways are still poorly understood in sponges. As you know, by the end of this century, the worst future scenario predicts that seawater temperature will increase by 3 degrees, pH will decrease by 0.3 unit, and dissolved oxygen will decrease by 4%. These extreme environmental conditions have been experimentally tested on sponge abundance, distribution, respiration, and symbionts, with contrasting results and species-specific responses. So some sponges are more tolerant than others. However, few studies have investigated the effect of climate change on sponge metabolism, then acidification, warming, and deoxygenation. To study the effect of sponge metabolism, to climate change on sponge metabolism, one of the best approaches is to use natural laboratories, where we can study natural variability and get ecologically relevant results. Among natural laboratories, such as CO2 seeps, macrotidal environments, and semi-enclosed lagoon, are natural laboratories where we can study the organisms' responses to ongoing climate change. Here, in New Caledonia, We have one of this natural laboratory, the Bouraké Lagoon. In Bouraké, organisms are exposed to daily fluctuation of pH, dissolvent oxygen, and temperature. And among coral reef organisms, sponges are particularly abundant. So this site was perfect for me to study the effect of extreme environmental conditions on sponge metabolism. Therefore, I divide my PhD into 3 aims. The first aim was to fully characterize the semi-enclosed lagoon of Bouraké, which is only shortly monitored. Then I perform a long-term physicochemical characterization and an assessment of the benthic community. I started these aims during my master in 2018. And the result of these aims highlight that one sponge in particular, Rhabdastrella globostellata, is the most abundant sponge in Bouraké. Then I use this sponge species as a study model for my PhD. The second aim of my PhD was then to understand the effect of extreme environmental conditions on sponge Rhabdastrella, with particular attention on sponge metabolism and photosynthetic activity. The result of these aims highlight that temperature could be one of the main drivers in sponge carbon change. Then, my third aim was to perform in aquarium an experiment to test the effect of only temperature on sponge carbon and nitrogen cycles and their incorporation in sponge tissue. Here I use sponge from Bouraké and sponge from a control site. I then organize my PhD presentation in agreement with these 3 aims. I explain by which each method has main result, and then I have a general conclusion and discussion. So, let's start with the Bouraké characterization. The Bouraké Lagoon is located in the southwest of New Caledonia, so here. And I also choose 2 control sites, R1 and R2, to compare environmental parameters. So to better visualize the Bouraké Lagoon, let's do a virtual trip together. So here is the entrance of the Bouraké Lagoon. We have a channel with a coral platform that's developed at the both sides of these channels. The outer lagoon is mainly characterized by soft coral and massive corals. Here, for example, we pass through an area that is rich in sponges, particularly my sponge Rhabdastrella. After that, we have a second channel, and we arrived in the first pool that is here on the left, where we can see the reef platform. After this first pool, we have a second channel, and also in this case, the coral platform developed at the both sides of the channel. And here we can see the branching corals. And after this second channel, we arrive at the innermost part of the lagoon, where also in this case we can see the reef platform, and is mainly characterized by branching corals. Here we have the view of the top of the semi-enclosed lagoon, which is completely surrounded by mangrove forest, and here we have the outer lagoon. So to characterize the seawater temperature, pH, and the dissolvent oxygen from 2016 to 2020, I used 21 stations in the white dot in the Bouraké and other stations in a control site. While to measure seawater chemical parameters in the 2 DL cycles in 2017 and 2019, I used 5 stations, the yellow square in Bouraké, and other stations in a control site, and I measure nutrients, organic matter, and seawater carbonate chemistry. To assess the benthic community distribution, I carry out in 2018 24 30-meter long transect along the Bouraké Lagoon, and each meter I took photo quadrats. I then analyze a picture to estimate the percentage of coverage of the main biotic and abiotic components. Thanks to the picture and also sample collection, I then identify the main benthic community, and I generate a checklist of the Bourraquet species. Here we can see some result of the physical parameters that I measured in summer 2020 in Bourraquet and in control site. We can see that Bourraquet have a daily fluctuation of dissolvent oxygen, pH, and temperature, while these parameters stay quite stable over time in a control site. So compared to a control site in Bourraquet, dissolvent oxygen can reach less than 50%, pH ranging from 8 to 7.2, and temperature could be 1 to 3 degrees more compared to a control site. So in Bourraquet, the physical parameters are driven by the semi-diurnal tidal phases, which generate two different environmental conditions. During high tide, we have close to normal condition, while during low tide, we have extreme environmental conditions. In addition, I also found that nutrients and total organic matter were more concentrated in Bourraquet compared to a control site. Among nutrients, silicates and total organic carbon were more concentrated during the low tide compared to the high tide. Looking to the benthic community distribution, I found some differences in the main biotic components from the outer to the innermost part of the lagoon. So the outer lagoon is mainly characterized by soft coral, followed by sponges in the middle, and branching coral in the first pool and the innermost part of the lagoon. So despite the extreme environmental condition of Bourraquet, I found a high biodiversity, and I identified 66 species of corals, 28 species of macroalgae, and 11 species of sponges. The aims here, in summary, was to characterize the semi-enclosed lagoon of Bourraquet using a long-term physicochemical characterization and an assessment of the benthic community. So both aims were completed. I found that physical and chemical parameters changing driving by the tide, reaching extreme environmental conditions during the low tide, that the geomorphology of the system seems to affect the benthic community distribution, and I finally characterized 105 species in Bourraquet. So these results are now published in Biogeoscience Journal and helped to recognize Bourraquet as a natural laboratory to study the effect of extreme environmental conditions on organisms. Then, this site was perfect for me to study the effect of extreme environmental conditions on sponge metabolism. For that, I used the sponge Rhabdastrella globostellata, that is the most abundant sponge in Bourraquet. This is interesting because this sponge is very rare to find outside Bourraquet and around New-Caledonia, so I only found the control site at the end of 2021. This sponge is known to be a high microbial abundance sponges, and also recent studies show the ability to uptake dissolved organic matter and bacteria from seawater. Then the aim here was to understand the effect of extreme environmental conditions on sponge Rhabdastrella, with particular attention on sponge metabolism and photosynthetic activity. So to study the sponge metabolism, I used the respirometric benthic chambers. The system consists in a chamber, hermetic chamber, where I put the sponge, a pump connected to a battery, a system of semi-flexible tube where seawater circulated, and a multi-probe that measures the oxygen change during the whole incubation. In general, I collect a sponge sample, I put the sponge into the chamber. At the beginning, at the end of 1 hour incubation, I collected seawater samples, I analyzed seawater samples, and I then normalize, and interpret data. I repeated this incubation during the two environmental conditions, so during a high tide and during low tide. And I also repeated in 2 years, so in 2018 and in 2020. Specifically, in 2018, I only performed a dark incubation using a set of 4 chambers with an empty chamber used as a blank. I repeated the 4 sets during high tide and 3 sets during the low tide. In 2020, I repeated this dark incubation using a set of 6 chambers, and I repeated 3 times during high tide and 4 sets during the low tide. This incubation allowed me to obtain information about the dark respiration, nutrient recycling, and organic matter recycling. In 2020, I also performed 20 like the dark incubation during high tide and during low tide, and this incubation allowed me to obtain information about dark respiration and photosynthetic activity. So, what did I find? Here we can see a box plot that represents the sponge respiration rates in 2018 and in 2020 during both high tide and low tide. Negative values correspond to our consumption. So even if sponges respire a little bit more in 2020 compared to 2018, I did not find significant differences between tides. So the sponge respiration rates is not affected by the tide. With regard to nutrients, I did not find significant differences between years, and the only nutrient that show remarkable differences between the tides is silicate, which was more consumed during the low tide in both 2018 and 2020 compared to the high tide. While the other nutrients did not show remarkable trends between tides. So the silicate cycle was affected by the tide. But the most important result is that the capacity to consume dissolved organic carbon, so the first step of the sponge loop, was confirmed during both 2018 and 2020, and during both high tide and low tide, with more consumption during the low tide. So the first step of the sponge loop was not affected by the tides. In 2018, we can also confirm the second step of the sponge loop, so the POC production. While in 2020, something interesting happens, because during high tide, we have a production of POC, then we can confirm the second step of the sponge loop, while during low tide, all sponges consume POC, and then the second step of the sponge loop was broken. To confirm this result, I also measured the real detritus production by subtracting the live particle organic carbon to the particle organic carbon. And similar to the POC, I found that detritus was produced during high tide while was consumed during the low tide. So definitely, the second step of the sponge loop was broken during the low tide in 2020. Here we can see respiration and photosynthetic activity. So I already showed you the respiration result. It's just to show you the photosynthetic activity that was affected by the tides with lower oxygen production during the low tide. So now the question is, why the sponge loop disruption only happens during the low tide in 2020 and not before? So to answer this question, I look into the environmental parameter between the two low tides in 2018 and 2020. The solvent oxygen, pH, and POC were similar between tides and years. Then by exclusion, high temperature and high DOC availability could be the main cause of the sponge loop disruption. My data show that only DOC reached double concentration during the low tide in 2018 compared to the low tide in 2018. However, my results also show that the sponge DOC uptake was the same during the true low tide, excluding the fact that high DOC availability could be the main effect on the sponge loop disruption. For the physical parameters, I found that temperature reached 5 degrees more during the low tide in 2020 compared to the low tide in 2018. Then I hypothesized that a temperature increase above a sponge-tolerant threshold and in combination with low pH and low dissolvent oxygen could be the main driver in the sponge loop disruption. In summary, the aim here was to understand the effect of extreme environmental conditions on sponge Rhabdastrella, with particular attention on sponge metabolism and the photosynthetic activity. So both aims were completed, and I found no main effect in the respiration rate, but a main effect on silicate cycle and carbon cycles with a disruption of the sponge loop pathway. I also found a negative effect on the photosynthetic activity, and I show that the sponge Rhabdastrella globostellata have the metabolic plasticity to cope with different environmental conditions. Finally, temperature in combination with low pH and low dissolvent oxygen is the main driver in the sponge loop disruption. However, I performed this experiment in situ, so this makes me impossible to separate all the multiple factors that can act in the sponge metabolism. These results are now published in Marine Pollution Bulletin Journal. Then, to understand if temperature is the main driver in the sponge loop disruption, I perform aquarium experiment. Here, the aims was to understand if temperature alone is the main driver in a sponge carbon and nitrogen change, cycle change, and also in their incorporation in sponges. So here I use a sponge from Boracay and sponge from a control site that I finally found at the end of 2021. Specifically, I have two hypotheses. The first hypothesis is that the temperature profile that I measured in Boracay in 2020 is the cause of the sponge loop disruption. So I perform an aquarium experiment using two different temperature conditions. 28 has in situ temperature at the control, and 32 as in situ temperature at Boracay. The second hypothesis is that the Boracay sponge have a specific way to recycle organic matter. So to confirm this hypothesis, I compare lab-laden coral mucus incorporation in both sponge from Boracay and the control site. So as I said before, it was really difficult to find the control sponges in Boracay and outside Boracay and around New Caledonia. And I finally found the control sponges in the east coast of New Caledonia, where Rhabdastrella reached 2% of coverage compared to the 40% of coverage the Boracay Lagoon. So in each site, a fragment of 50 different mother colonies were cut and allowed to restore in situ for 3 weeks. In Boracay, I also collected a coral sample for coral mucus labeling. Then I collected sponge and coral samples, and I acclimated them in aquaria from 1 week, for 1 week. Corals were then labeled for 5 consecutive days, 8 hours each day, using sodium bicarbonate and sodium nitrate. Then 6 sponge aquaria, containing 8 sponges each tank, 4 by each site, were kept below the coral aquaria to allow the transfer of coral mucus from the corals to the sponges. 3 of them were maintained at a controlled temperature of 28, while 3 of them were maintaining to the fluctuating temperature of Boracay from 28.5 to 33.2. So after the transfer of coral mucus into sponges, sponges were then incubated for 3 hours at 28 degrees and 32 degrees in agreement with the temperature treatment. At the beginning and the end of incubation, I collected seawater sample for organic matter analysis, and I also collected sample to investigate the coral mucus incorporation into sponge tissue and detritus. I repeated this incubation also for sponge maintaining in a control condition, then without the adding of labeled coral mucus, but maintained at the same temperature treatment. So here we can find the first main result at the level of the sponge carbon cycle. So we can see that sponges from Buraké and sponges from control site during both 28 and 32 temperature consume dissolved organic carbon, even if with high variability, and lower DOC consumption during at 32 degrees. So we can confirm generally the first step of the sponge loop. While if we look into the second step of the sponge loop, that is the detritus production, we can see the sponge loop pathway for the Buraké sponges at 28 degrees and some control sponges at 32 degrees. So here, again, the second step of the sponge loop was broken at 32 degrees, so we found the same results that we found in situ. To confirm this result, we can also look into the incorporation of carbon-13 and nitrogen-15 into sponge tissue and detritus. So Buraké sponges showed higher incorporation into tissue, while control sponges showed higher excretion of carbon into detritus at 32 degrees compared to the 28 degrees. The same trend was also found for nitrogen. So we can say that for both populations, a temperature of 32 degrees causes a change in sponge carbon and nitrogen recycling. But in which direction? So to answer this question, we can look into the tissue and detritus ratio of carbon-13 and nitrogen-15. Buraké sponges increase the carbon-13 ratio from 1.6 to 2.4 at 32 degrees, while the ratio of nitrogen-15 increased from 2.1 to 3.1 at 32 degrees. So the Buraké sponges increase carbon and nitrogen incorporation into sponge tissue, while control sponges have an opposite response because they incorporate more carbon and nitrogen in detritus, leading to a reduction of this tissue and detritus ratio. Then Buraké sponges have a different response faced to higher temperature and compare to control sponges, and maybe have the better strategies to cope with higher environmental condition, higher temperature. So if we return back to our hypothesis, both hypotheses were confirmed, because I found that a temperature of 32 degrees causes a disruption of the sponge loop pathway, and I also found that Buraké sponges have a different pathway to recycle organic matter compared to the control sponges. So in summary, the aims here was to understand the effect of temperature to carbon and nitrogen cycles and their incorporation in the sponges. So both aims were completed. I found that both sponges uptake dissolved organic carbon released by corals, that a temperature of 32 degrees causes a change in the sponge carbon and nitrogen recycling, with Buraké sponges that increase their incorporation into tissue, and control sponges that increase their excretion into detritus. So these are the main results that I found during my thesis. But when I started my PhD, my first question was, how is possible that Rhabdastrella is so massive despite the extreme environmental condition of Buraké? So now I use my finding to answer this question. So I show you that the main metabolic change were at the level of silicate cycles and carbon cycles. So silicate were more consumed during the low tide in both 2018 and 2020. So we can hypothesize that sponges uptake more silicate to build a more robust skeleton made by a more number and longer spicules, And this flexible and robust skeleton could help sponge to stay healthy and grow despite the extreme environmental condition. The second main change was at the level of carbon cycles. So during normal condition, I show you that Burack's sponge do the sponge loop pathway, which is a different pathway normally found for massive sponges for which low detritus production was found. So these different strategies used by Burack sponges could be due to the specific environmental condition of Burack. So the high-rich nutrient environment of Burack could help a sponge to maintain a high cell turnover and renewal. During extreme environmental condition, I found a disruption of the sponge loop pathway, then a similar pathway that massive sponges, but Burack sponge have a different pathway compared to normal condition. So this changing pathway could be a strategy used by the sponge to cope with extreme environmental condition and then save energy. So I believe that the sponge Rhabdastrella is adapted to the continuous changing tide and the environmental condition and adopt the best strategies according to the tide to have the best fitness and then save energy, stay healthy, and become massive as I observed. I always talk about the extreme environmental condition, but I also show you that temperature is one of the main drivers in a sponge carbon change. So if you look into the summer temperature in Burack, and we choose 30 degrees as a temperature threshold for the sponge, sponges were exposed to 10% of the time at temperature higher than 30 degrees. If you look into the warmest day during this summer period, sponges were exposed for 30% of the time at temperature higher than 30 degrees. Then, the repetitive exposure to a higher temperature stressful condition could help sponges to develop mechanisms to reduce cellular damage and then stay healthy and grow despite the extreme environmental condition of Burack. If we now compare the responses that I found in control sponges and Burack sponges, that was only possible for me in ex situ, so in aquarium, we can see that at 28 degrees, control sponges do the biomass pathway, while Burack sponges increase detritus production. So we have two different pathways, the biomass and the sponge loop pathway. Because the environmental conditions were the same, this could reflect an environmental adaptation to the origin of the sponges. And then also I liked different ecological traits. If you look to the response at 32 degrees, we have an opposite response because the control sponges increase detritus excretion. So suggests that the sponge underwent to stress temperature, so higher cell degradation and then high detritus production. While Burack sponges increase carbon and nitrogen incorporation into tissue. Then the different response faced to higher temperature could reflect a different tolerance faced to higher temperature and then also a different thermal history of the two sponge species. Burack sponges seem to have the best strategies to cope with higher temperature and then save energy and prevent cellular damage, a preventing mechanism that control sponges seems not to have. So, if we return back to my first question, and I think that the massiveness of the Rhabdastrella in Burack is mainly due to the specific characteristic of Burack. Then, the rich nutrient environments of Burack could help sponge to cope with extreme environmental conditions, and also the fluctuating environment of Burack could increase the resilience of sponges under extreme environmental conditions. Then a sponge develops metabolic plasticity to cope with the different environmental conditions and to adopt the best strategies according to the tide. Then a sponge save energy, grow, stay healthy, and became massive as I observed. So in conclusion, I use 3 different approaches to understand the effect of extreme environmental conditions on sponge metabolism. I show that Boracay Lagoon is a natural laboratory to study the effects of extreme environmental conditions on organisms, and particularly here on sponges. I also bring the first insigne into the effect of extreme environmental conditions on sponge metabolism, with a main effect on silicate cycle and carbon cycles, and then a disruption of the sponge loop pathway. And finally, I found that a temperature of 32 degrees causes a change in sponge carbon and nitrogen recycling, with a possible cascading effect on the whole coral reef. Then, in a world where climate change is accelerating and the effect of climate change on sponge metabolism have yet to be studied, my thesis brings one example of how tropical sponges can cope with extreme acidification, warming, and deoxygenation. I show that the sponge Rhabdastrella have the metabolic plasticity to cope with different environmental conditions, adopt the best have the best fitness according to the tide, and suggesting that the survival of sponges and possible other taxa will be possible in future. However, this could lead a change in sponge metabolism and then also in the food quality and availability in the whole coral reef. So to go beyond the scope of my thesis, but try to understand the sponge metabolism, I also look into the carbon budget in Boracay sponges. So I show you that during high tide, the sponges show a lower respiration and lower carbon uptake, and during low tide, the sponges have higher respiration and higher carbon uptake. So the carbon budget in sponge could be calculated using the respiration quotient, that is the CO2 produced by oxygen consumed, and I found an average of 1.2 during high tide and 1 during the low tide. I also calculate the IR index, that is the ingested carbon by the oxygen consumed, and I found a result of 0.2 high tide and 0.8 during the low tide. When this ratio is lower than the respiration quotient, sponges have not enough energy to compensate the respiration cost. This difference was 1 during high tide and 0.2 during low tide. These lower differences during low tide, I think, is due to the rich nutrient environment of Boracay during the low tide, but remain unresolved where carbon comes from to compensate the respiration cost. So one hypothesis could be at the level of bacteria symbionts. They could be divided into phototrophic and heterotrophic symbionts, and both have normal activity during the high tide, then can provide the extra energy to the sponge to compensate for the respiration cost. While during the low tide, I show that the phototrophic symbionts are negatively affected, while heterotrophic symbionts show a normal activity. So heterotrophic symbionts could provide the extra energy to the sponge for the respiration cost, and hypothesize that heterotrophy could have a higher implication in the sponge metabolism to cope with the extreme environmental conditions. So these are only hypotheses that open future questions. So what are the next steps of my thesis? I have a lot of next steps. But I would like to answer some questions in my future project. So the first thing is that I work on one sponge species, and there are more than 8,000 sponge species all over the world, both massive and encrusting, and both high microbial abundance and low microbial abundance. So I would like to perform a future study on understand or increase our knowledge about carbon and nitrogen cycles in sponges, and also understand the effect of climate change on these cycles and have a more generalized responses. I told you that sponge is not only a sponge, but is in association with microorganisms that could play a very important role in sponge acclimatization and adaptation in a changing environment. So I would like to perform a future study on the assessment of the microbial community in both control sponges and boracic sponges, and also understand the heterotrophic and the phototrophic contribution to the carbon and nitrogen cycle in sponges. I also show you that when carbon is not released as detritus, is mainly stored in sponge tissue, and hypothesize that is used by the sponge for the biomass. So I would like to perform a future study to answer this hypothesis by performing some biomass measurements and also using biochemical label, such as a stable isotope analysis, followed by a nanosims technique to understand and better visualize where carbon is really stored in the sponge. In addition, I also would like to characterize the carbon source in boracic, because boracic is a really particular site, and so the differences in the carbon source could also lead to a difference in the carbon sponge pathway. Finally, I really would like to understand if the response I observed is the result of adaptation or metabolic plasticity. Unfortunately, I found the control sponges only at the end of 2021, so this makes me impossible to further analyze and understand the ecological traits of the sponge. So I would like to perform a future experiment, such as a cross-transplanting experiment between control and boracic sponges, and do some transcriptomic analysis and heat shock protein analysis to understand if the responses that I observed is the result of adaptation or physiological plasticity. Before finish, I would like to thank all the contributors of my thesis that make the realization of the PhD possible. So thank you to all of you, and also thank you all of you for listening to me.
So I will not repeat the congratulations that I formulated in my report. In general, I like very much that kind of research work based on the realization of experiments here, both in situ and in aquaria. Well, it's true that you benefited from a quite exceptional experimental site, which really gives you a great desire to explore. So I wish one day I will be able to come and see that. So these conditions of realization of this research have already allowed you to value your work by very good publication. And I guess that you have already received a lot of criticism on your work by the reviewers of these articles. Having said that, as I have written in my report, in general, I think that your work has not been based enough on control situations. So therefore, outside the boracic site, so perhaps we'll discuss that a bit later. And so as we can always do better in a thesis, and this is the things that you could have done differently that we are going to discuss a bit. Perhaps in order to better know what you will do next, but you already mentioned a number of wishes in your final slides. I could see that you took into account some of my suggestions, comments, and I'm very happy of that. And perhaps we will finish with a few questions about what you would like to do in your future research. Well, I kind of start without a first comment on the way you presented your sponge taxonomy. Well, you understood that I was not very happy with the way you presented the spicule preparation and the lack of some details about that. It's a bit frustrating for me. But as you mentioned in your result, that the silica uptake was affected by the tides, I would like to know if you really checked the difference that you can have in a species composition between the Rhabdastrella in Boracay and Rhabdastrella in control sites. Did you find any phenotypical traits changing between these two conditions in terms of species composition, size, for instance?
Thank you. Okay, thank you very much. Yes, about speckles, I just observed the differences between control sponges and the Boracay sponges, and actually I found that Boracay sponges have more number of speckles. So I have not time to look into the length or the width of the speckles. But I'm pretty sure that some things happens. So I really would like to go further also on the identification about this difference at the spedicle levels, but also genetic levels.
But in Rhabdastrella especially, well, there is a huge collection of speckle types that may be affected in their shape or size because of the silicate availability. So did you see anything, or did you try to see anything?
No, I'm sorry, but I didn't look into these details. I just use a speckle for confirm that it was Rhabdastrella. So I just just check that I have the impression that I have more number of speckles, but I think that what I expected is to find more the speckles which are more width and also in the size more bigger.
And do you know anything in the literature about this relationship between a speckle type or size and availability of silicates?
I know that the increase in the silicate availability and then also used by the sponge for build the skeleton could help the sponge to have more flexibility and also could be more robust, could help the sponge to there are recent papers that show that could help to contrast the extreme warming and acidification. And also, I think that could be a good strategy for Rhabdastrella because in Boracay there are a lot of current. So also this flexibility that could have with this high number of speckles could help the sponge to stay in this site.
Okay. Well, you have some works by Paco Cardenas in a group close of Rhabdastrella. That is trying to correlate this availability in silicates with the change in size of some speckles composing some geodia in the deep seas, especially. So he's working on bathymetric gradients and assessing these putative changes. And in the same way, I was a bit surprised to see that you will be. You had measured the photosynthetic activity without any description of the microbiome of your sponge model. So are you sure that Rhabdastrella has some cyanobacteria, for instance, or photosynthetic symbionts? How far do you know? Do you have a basic knowledge of the microbiome composition in this group, at least? Genus, maybe you could have some reference from different other species.
Yes, thank you for this question. So I would like to share. I can share. Yeah. So thank you for this question because this is I investigated the microbiome of the sponge Rhabdastrella, and the only paper that showed the Rhabdastrella microbiome is the paper of Steiner, and we found a really interesting result. Because, I try to show you better. Really interesting result. Okay, here is another sponge species, but if you look into Rhabdastrella, is mainly composed by chloroflexi, and interestingly, we have that cyanobacteria represent 10.6% of the microbial abundance. If we compare this with normal Rhabdastrella found in Pacific, that only reach 2%, I think that there are something in cyanobacteria in the Rhabdastrella of Buraque. And this could be very interesting to investigate more, because unfortunately I have not a lot of sample here. So I need to confirm this result also with a more number of samples. But chloroflexi could have a really important role in Rhabdastrella at the level of photosynthesis and also nitrogen fixation.
Okay. Well, another question about acidification. In your introduction, when you're talking about acidification and its putative effect on marine organisms, you mainly cite a review, a chapter in a book, Gattuso, I don't remember the second name, but it's a very good chapter. I fully agree with that. But I would like to know what, if you have any precise examples of acknowledged effects of acidification on marine invertebrates and especially on sponges. What did you see in the literature?
In general, or at the level of carbon?
Well, let's talk about effect on the metabolism or skeleton genesis.
Okay, so there are really different opposite responses based on species. So I found what I remember is that there are some species that were more tolerant under ocean acidification, and other that were less tolerant. So we have the so-called winners and losers in a future coral reef. I think that I can do a general answer, because there are really species-specific response that depends on the geographical location and also sponge species.
Yeah, but so this comment that this answer that you gave me is related to one of the comments that I wrote in my report. Is in general, it's very hard to give a general answer when talking about a phyla like Porifera. You would find well, very, very variable responses, and it's true that most of the papers that were published on this question about acidification and its effect on sponge metabolism are not very congruent, and actually there is a majority of papers that are not showing anything really, except that one with exactinylate, so deep sea sponges, and true effect on their pumping activity and skeleton genesis. So in this case, this is something that you can relate to your work. Then you know that I have a very heavy question that it asked you already when we were in Leiden together. So it's something that you present in your chapter 2. And that you didn't discuss. So I would like to have your opinion, because I don't have any answer to that question yet. How do you interpret the huge variability in terms of stable isotopes variability that you found in sponges?
Yes. Also here I have something to show you, if I'm able to show you. This one, yes. So it's a little bit smaller, but yeah. About the stable isotope in sponges, I think there are some interesting things to say. So again, unfortunately, I have not a lot of samples, so it will be good to repeat these experiments with high number of samples. But the interesting things is that I have differences from the outer to the innermost part of the lagoon, because here, for example, the orange is the innermost part of the lagoon, while the violet is the outermost part. So we see a really difference between the carbon and also nitrogen incorporation. And I think this is, so we have more negative carbon 13 and also more negative nitrogen 15 inside the Boracay Lagoon compared to the outer lagoon. And I think that this is lead also to the specific topography of Boracay. So it's a mangrove, and there are a lot of rich also terrestrial inputs. And then the sponge outside, more inside the Boracay Lagoon have access to more terrigenous source. And also the fact that I show chloroflexi and cyanobacteria also could explain these differences in carbon and nitrogen. And from the outer lagoon, so here, they are on higher nitrogen 15 and carbon 13. I think that is mainly due to the fact that they are more near to the outer lagoon, so of renewal of water, and they can food more in marine lagoon source. So this, unfortunately, really, I have not enough data to say something strong, but yes, is a preliminary of observation.
So you are mainly explaining that by the origin or the location of your samples, but well, you know that because we discussed that together. But I got exactly the same kind of variability with different sponge species that were sampled in a given site, exactly the same site, and a very huge variability with no true pattern that I could explain. So in this case, we cannot explain the location. By the location, we can explain that by a putative phylogenetic signal, because you can see that you have different species belonging to different taxonomic group everywhere in the diagram. So what could be the other hypothesis to explain that variability? I will precise my question. What do you know about the variability of that kind of signature in prokaryotes?
I can't answer you, but is a things that I have to investigate further. The two hypotheses that I made is only at the level of the different carbon sources from the outer and the inner, and also possible different microbial community. So yes, I have to investigate further.
Yeah, this is what I read, and what I was willing to say is that it may be related to the symbiosis and the composition, not in terms of abundance, because again, if you try to opposite HMA, LMA, you cannot find a true pattern, but perhaps in the microbiome diversity, there may be an explanation, and something that would be very interesting to understand, because, well, it's well known that these symbionts are playing a key role in the uptake of this organic material.
Yes, I totally agree, and I also think that if we look to the microbiome composition of the same sponge species in the outer lagoon and the inner lagoon, we found differences. We can find differences.
Okay, so let's go on with a sponge feeding strategy. And again, but a little criticism on the way you presented your work is that you didn't use enough all the literature, what we know very well about sponge feeding strategies and how variable again it may be when you consider the world diversity of the phylum Porifera. And I want to stress a bit on the fact that the sponge loop concept that you are working in your PhD is coming from a very, very, very peculiar example of a sponge feeding strategy. So what do you know? What I could see that you know some things about the model that use Jasper de Gouges and his collaborator in this description. But what do you know exactly about the I would say the weird biology of the sponge species that he used to describe the sponge loop?
Yeah, the sponge loop pathway was described in encrusting sponges and mainly in Alisarca caerulea, and so is a completely a different sponge species as my Rhabdastrella. I know that there were some study about the cell renewal of these sponges that take up more or less 6 hours.
So yeah, tell me a bit more about this. Tell me what is this cellular renewal? What do you know about this? Tell me a bit more about this. What kind of cells are we talking about?
The sponge cell. So
yes, the sponge cells. But no, but I precise and you go on. The sponge cells that are renewed a lot by Alisarca caerulea are choanocytes. Okay, that's cells that are responsible of the particle uptake. And because this sponge has a very high dynamic, it's a fast-growing species, an encrusting species that is a true competitor. Yeah.
They do not have to compete for the space, so they have to investigate. They have to compete for the space, so investigate in this to maintain their cell renewal. Like rejuvenation of the cell.
And the number of choanocytes and the size of the choanocyte chambers are just huge in this species. So that explains that special cell dynamic in this species. So it's not a surprise that this species will give a picture of DOM assimilation, a balance that is in favor of a DOM assimilation by the sponge and a huge release of particles that are actually cells, choanocytes that are renewed because they are very, very abundant in this sponge species and that has a very high dynamic. But in general, if you had to define the sponge feeding, like in a dictionary, on an encyclopedia, what would you say? Would you say that would you use the sponge loop to define what is the sponge feeding strategy, or would you define this in another way?
Do you mean R and K strategy?
Yeah.
Okay.
If you had to give an average feeding strategy for sponges,
I think that the most sponges are R strategists.
Are what?
R strategists.
I didn't get the answer.
R strategists. R strategist.
I don't understand.
Stratégie R, Thierry.
No, I'm not talking about R or K strategy. I'm talking about the feeding strategy. So are they feeding on dissolved organic material mainly, on particulate organic material mainly?
For Alisarca is dissolved organic material, and for a lot of sponges, the diet most consists in dissolved organic materials. Well, for example, I found something interesting in my sponge. So I think that during the different tide condition, I could have a different feeding also in the proportional of dissolved organic matter and particulate organic matter. So this is a thing to investigate more. But globally, they are dissolved organic matter feeding.
Okay. And when you talk about dissolved organic material that are released, you're mainly considering this DOM as a source of food for other organisms. Okay, when you talk about the sponge loop in coral reef, especially, but don't you think that that DOM can have a other functional role in the ecosystem functioning? And what do you know about this?
I know that, for example, there are also bacteria that feed on dissolved organic matter, so can make this microbial, the so-called microbial loops. There are what to talk about? There are yes, I think that the most of organisms feed on dissolved organic matter.
Yeah, okay, I agree with that. But well, you consider DOM in all your discussion like waste, like a product of the catabolism of the digestion by your sponge species, and I believe that that DOM may have other functional roles in the ecosystem, because we have some literature about this, and I must tell you that I'm working on that. So what can you say about this?
You talk about DOM or POM?
DOM, yes, DOM,
okay.
Yes. There are a lot of paper by people in the States, Carafiore, for instance, or in Marseille, very good paper, I would say, showing that sponges are able to release some dissolved organic compounds that are used for chemical mediation, communication between species. And it's not a question of waste. They are just biosynthetic products that are involved in the communication between sponge species and other organisms. Okay. Well, you did mention a number of questions that I wanted to ask you, especially what will provide you to work on two contrast ecological strategy. So using species of R and K strategy. So you did mention that, so I want to ask any question about this. So maybe to finish, you mentioned that well, you were discussing about the fact that your sponge species is saving energy to better cope with this environmental change that is provoked by the tides or that simulate the climate change. I agree. There should be a trade-off between primary functions and secondary functions. This topic also we have been working a lot here in Marseille, but how could you better answer this hypothesis? What kind of strategy would you implement to better answer this strategy of trade-off when there is a change in environmental conditions?
Yes, but I think that they decrease the cell shedding, and that use this energy to build biomass, maybe like simply increase the size of a conocyte. So try to invest this energy in a different way than just renewal cell from proliferation and shedding.
Okay, why not? And my very last question, but this could be a very long discussion. Well, it's a bit frustrating, but again, what you did was already so great that we cannot be too strong with that. But of course, when you are when you are working in such a crazy places like a Boracay, you could expect to see some work about adaptation, true adaptation, assessment, capabilities assessments, and so yes, if you had to repeat today this research, well, you did mention some perspectives, but if you had to change the design of your experiment, what you do try? What will be your priority if you had to start again your PhD and do it in a totally different way?
The first thing is that I would like to generalize my observed responses. So using more different species and understand how these species, if you said about Boracay, so if you, for example, we use different species from a control and in Boracay and do this cross transplanting to see if we have, if also control sponges have the ability, have this metabolic plasticity to cope with this environmental change, that we can exclude this possibility. And also go more deeper, so like using, for example, as I said, the transcriptomic analysis for understand if really there are genes that regulate the adaptation of the sponge inside Boracay to the Boracay conditions.
Yeah, I fully agree. The great disadvantage that you have when you want to work on adaptation with sponges or coral is that they are long-living species. But the great advantage is that they are cloning organisms. And what will be very easy is to have different transplants of a given genotype, so you can work on much more than plasticity. You can truly work on adaptation. And yes, if I had to do your PhD, I will start with that, I think. But anyway, it was very, very good. I was very, very happy to read this work, and well, congratulations again, because you did a great job in publishing most of your results. I think you have many more because as I wrote in my reports, you have a lot of data in your appendix that you should better use for further research, perhaps, or perhaps for new papers. So this is a very it's a very high value, and I'm pretty sure that you will do you will become a great researcher in the future. So thank you again.
Thank you very much, Thierry.
Okay, so thank you, Thierry, and thank you, Federica, for answering the question. So Mathieu, could you please address remarks and comments?
Thank you, Claude. Thank you, Federica. Can you hear me okay?
Yes.
Great. I'm going to start by congratulating you and your supervisor. Same as Thierry, I was very impressed by your work. You know, I really enjoyed it. I found it very rich, the thesis, and this presentation was very clear. What I enjoyed the most was the fact that you did a lot of different things. You know, that would have been challenging, I would imagine, for a PhD. And everyone here who has done a PhD knows about that. You know, you really did some work at the interface between ecology, physicochemistry, and physiology. So integrating a lot of different approaches. I also respect and really enjoyed the fact that you did some work on site and experimental work in situ and in aquaria. So you really did see a lot of the challenge that we have in research in general. So you've done it all in 3 years, I would say. So, and that's credit to your supervisor, I think, because when we read the PhD, we can really see this transition from the ecology and the ecosystem of Boracay to the in situ experiment and then the aquarium experiment. So well done, really, with that. I really enjoyed that. So congratulations again on that. I'm gonna have. I'm not an expert in sponge per se. I'm more. My field is more symbiosis, microbial symbiosis, host microbe symbiosis, coral reef, a bit of sponge and seagrass. So I'm gonna be maybe a bit less specific than Thierry, but I agree with most of his comments. I might have a bit less question. I'm gonna have one first question about the model, and then I will try to follow with some question about the different chapters in the order, and then finish off with maybe one or two questions about what's next for you in the same way as Thierry. The first one, and I know we've kind of touched on this with Thierry and in your PhD, and even during your presentation, I saw that you tried to address that, but really sponges are massively diverse group, and there's a lot of things that affect potentially what you see here. The taxonomy, low versus high microbial abundance, cellular structure, feeding. There's a lot of things there. So I was wondering if you can answer if you think that Rhabdastrella is a good representative for sponges in general, a model organism for sponge, and if yes or no, what makes you think if it's a yes or if it's a no?
Yeah, thank you very much for the question. So I don't think that Rhabdastrella could be representative of the wall of sponges, particularly Rhabdastrella from Buraque, because it's a really particular side, as I described. So, and also, when we compare the sponge with the control sponge, that yes, I have to investigate more, because I think that we have to start with the ecological traits of the sponge outside Buraque to understand what happens in the sponge, in Buraque sponges. I said no, yeah, because I think that we have more to study. If we want to use this sponge species, we have to study also what happened outside Buraque, and maybe also in other natural laboratories. Why not?
Thank you for your answer. And I saw in obviously your future direction that you really try to address that by saying, yeah, we need to look at a multitude of species and things like that. So I know you're aware of it, but yeah, it's important to remember that Rhabdastrella is very abundant there on this site, which, by the way, I did dive there, and it's an amazing site. Buraque is pretty amazing, but yeah, it might not be representative of worldwide and other sites. So that's important to be aware of that. I'm going to go through chapter 2 now, and a bit more into photosynthesis. So I enjoyed the fact that you showed us some extra slides just right now with a bit more about the microbiome. I had a question about the photosynthetic symbionts. So great to see that it's actually some cyanobacteria. Something I want to point out, and it's before a question, it's more a comment. We have done, that's the kind of work that I do in sponge, is actually to look at the translocation of carbon between the symbionts and the host. And we've seen clear differences between, if it's a cyanobacteria, for instance, and dinoflagellates or other algae. Very clear difference in the translocation, so how much carbon is going from the symbionts, the photosynthetic symbiont, to the host. So be aware of that. That's number one. The other thing is cyanobacteria are a bit quirky and weird in their photosynthesis. So that's another thing to be aware is some of them can have different chlorophyll. And therefore, when you study their photosynthesis, you might want to be aware of that and do it differently potentially. Sometimes, even doing pump fluorimetry, you might have to use a different spectra of light to look at the photosynthesis of the cyanobacteria compared to the dinoflagellates. Anyway, that's a comment. Now getting into the question for this chapter 2. In terms of photosynthesis, you look at the photosynthesis at high tide and low tide. I was wondering, it was not clear for me when I read your PhD and in this presentation, how did you control the effect of the time or the different light level? Because obviously, yes, it's low tide, it's high tide, but that can occur at different time during the day. And obviously, the light penetration is not going to be the same. So looking at photosynthesis at high tide and low tide, you will have a lot of other factors involved there. So I just wonder if you tried to control that or you just did what you could.
Yes, I totally agree with you because I have really differences between high tide and low tide, also at the level of the light that touch the sponge, that arrive to the sponge. And what we try to do is to do in inverse procedure during also we have differences in the morning and the afternoon. So what we can try to do is to also inverse the dark and light incubation to try to have the same light during the morning and the afternoon and during the same high tide and same low tide. And another things, and I'm sorry, I not have the result here, but we also repeated this incubation in a laboratory. So using the same light for both high tide and low tide, but using the seawater characteristic of Buraké during high tide and low tide. And we found a same result with a lower oxygen production during the low tide. So it seems likely that symbionts are affected by the combination of low oxygen, pH, and temperature.
Great, thanks for the answer. And I saw, I was positively surprised by the fact that you improve your experimental design with the time. I could see that the sampling size in 2018 was not equal in the different group, but then 2020, you improved the sampling design and had 20 replicates in each group. So yeah, I know what it is to work in the field, and I know how challenging it is. So yeah, it's important to be aware of all of this. Something to kind of link this experimental study with the chapter 1, where you characterize Buraké. Buraké sites and the different organisms there and groups. Do you think there is a possibility that with these two different it's kind of two different worlds there during low tide and high tide on these sites because of the geology of these sites? It's a very different and extreme change. Do you think low tide favor different ecological group in Buraké compared to high tide? Is there a possibility that based on the group that are more photosynthetic versus heterotrophic, their performance is better at high tide and the opposite happen for the other ones?
Yes, this is a possibility because because I think that during the low tide, the heterotrophy gain compared to the phototrophic. And so all the organisms, such as also corals, for example, need to have a change in metabolism to shift between heterotrophy and phototrophy. So beside the benthic organisms, I think that it would be also differences in some, for example, fish. So there are some fishing that enters during the high tide and that go out during the low tide. But this is it's really something that needs to be investigated more.
Yeah, indeed, because I mean it's that will explain also why this site is so diverse. You know, with having in some ways two sites in one. You know, it's so extreme the change that occur there between low tide and high tide. Okay, I think moving to chapter 3, you saw different responses in Boraké sponges compared to the control one, different response to temperature. You went into it a little bit with Thierry, and obviously, you know, the one thing that could explain that, and I'm going to dig into that because that's where I'm coming from. It's not the only reason, but one possibility is the microbiome, you know, different microbiome. Now, I'm aware of a fair bit of work that has been done on coral, different species of coral in a similar way, with actually some translocation experiments with the coral from Boraké. So I'm not going to say that coral are the same as sponges because they are quite different, but purely from the holobiont, you know, they have a microbiome there. What do you know from this work on coral in Boraké? What does that show? You know, do we see different microbiome outside of Boraké that could explain why this coral, in that case, I'm looking at the literature on corals only here, are more tolerant? And do you think you could apply that potentially to the sponges?
Yeah, so microbiome analysis on corals in Boraké still on work, and the only preliminary information that I have from colleagues is that there are some Simiodinium inside corals in Boraké that specific clade that could help corals to cope with bleaching. So yes, I think that microbiome could be one of the main answer of these differences in population in Boraké and outside Boraké.
And what would be, sorry, I'm diving a little bit more into that, but and it's a link to the future also, but if we were telling you, okay, we give you funding for 1 year, to disentangle that, we fund your postdoc. How would you do that? Like, how would you, if you try to answer if it's a host factor, symbiont factor, that explain the tolerance, let's say just to temperature at this stage, what would you do? What type of experiment would you do to do that?
Yeah, like for example, we can just assessment the difference in microbial community during high tide and low tide, or if we are focused on temperature, so it's difficult to do this. Maybe we can do differences also between summer and winter seasons, and also well, here is just a preliminary experiment that I would like to do, but it's just using, for example, here on sponges. Sorry, it's not this one. It's this one. But likely use 2 different temperature and 3 different sponge species, and see in aquarium. So after a cross transplanting from Boraké and the control sponges, put them in aquaria at 2 different temperature conditions and make an assessment of the well, beside the carbon cycles, also in the microbiome. So looking into differences if we have some bacteria that increase or not in a higher temperature condition, and that could help the sponge to cope with the extreme environmental temperature.
Yeah, that's really interesting, and I like the design. The only thing I would add, and that's similar to Thierry, I would definitively include some genotyping and some rad sequencing or things like that to make sure that you have, you're looking at the same, population potentially, because you might have a lot, even within the site of Boracay, you might see a lot of differences there. So you will want to control for that. So, make sure that you genotype the host and the symbionts, and then, yes, translocation experiment would be a good way to kind of answer that. But controlling those factors, those genotype of the host and the symbionts. Maybe something moving towards, I'm mindful of time, moving towards opening up a bit more about what to do next. I really enjoyed in your presentation the fact that you had those perspectives, and you had at least 4 or 6 different directions. I was wondering, again, similar to the question I asked, although they are constrained what type of question you would address. If we were telling you, okay, we give you funding for a year or 2, which of these directions that you listed at the end of your presentation would you do in priority? And I'm saying that there's 2 questions in one question there. Number one is what is the most important question in the 4th or 6th direction that you gave at the end of your PhD, number one. And number 2 is maybe more personally what you would like the most to do, what you feel is more your little garden.
Okay, thank you for this question. So I think that what I would like to first answer is if really we have an adaptation of metabolic plasticity of the sponges in Boracay. And after that, I really would like to understand the change between sponge host and all the associated microorganisms. So this, like for example, as you know, also the nanosim techniques could be also a good technique to use to visualize where carbon is stored in my sponge, and also maybe also use some technique like fish technique to understand the exchange between the sponge and the sponge host and the microbiomes.
Okay, and what do you enjoy the most? Like, would you like to? Because obviously, what I see from this PhD is that there's a lot that could be addressed by looking at the microbial side of things, but is this something that you are interested in personally?
Yes, of course. Also, yeah, I'm interested in a lot of things, but yes, also in microbiomes, because by my preliminary results, I find something interesting. So I would like to understand what happens at the level of the microbiome.
Okay, because I'm asking because some people, don't really want to touch microbiology, so it's always it's always a question to. It's not only about the scientific interest, but also about the personal interest, and maybe even broader than that. Where do you see yourself next? Do you see yourself in academia, or do you want to continue in research and academia, or do you want to, I don't know, to go more towards some form of ecosystem management and things like that?
I really would like to continue in research. So I'm really happy if to share my postdoctoral project with all of you. If you have other ideas and to increase this my little dreams. So yes, the first objective I think is to go further on this direction and become a researcher.
And to continue on sponges, sorry to dig a little bit more into that, or would you be keen to open up to other organisms?
I open to other organisms. Also, if sponges stay with me for 4 years, so I'm a little bit attached. But I'm really open to also work on other organisms. Also, because in my team I have
I can see Thierry is reacting to that. So that's
yes.
Okay. Look, I'm going to end up here because I'm mindful of time. I just want to congratulate again you and the team behind this. There's always some challenges, but you have achieved a lot, and very rich work. As Thierry mentioned, I think there will be more paper out of that. So make sure that you value all the data set that you have as much as possible. You know, it's an important one, especially if you want to continue. That's why I was asking if you want to continue in an academic career. That's very important. You know, that's your currency. So well done again, and good luck for the future, and we will be in touch.
Thank you very much.
Okay, thank you, Mathieu. I can guess that you offer postdoc to Federica. Do you? Okay, so we can follow, and now I will continue with Verena. If you want to take the floor, please.
Yeah, hello everybody. First of all, I would like to apologize for my voice. I hope that you can still understand me, but I have a cold, so it's really affected by that. I would also like to really congratulate you, Federica. This was a really impressive thesis and a very nice, clear presentation. And even though I mostly work on coral, I work in similar environments. And so a lot of the work that I do with my team is quite similar. So I'm very familiar with the challenges and of working in these environments. And I think, like many of the others have also already pointed out, it's really nice, diverse thesis that shows a lot of different techniques. So congratulations on that.
Thank you very much.
I would first like to ask you a few questions that are a bit more specific to the 3 chapters, and then kind of come back to the more general concept of the natural laboratories. So for the first chapter, the benthic and abiotic characterization, I found it really interesting to hear that the sponges are most abundant in the middle section, but not so much in the inner section where the corals dominate. So it seems that they're quite resistant to these extreme conditions, but how do you explain that they primarily occur in the middle section?
Sorry, I don't know if I understand well. You said if the differences of the distribution of corals in Boracay, is that?
No, actually more so the sponges. So I think I remember you showed that they are mostly abundant in the middle section of the lagoon, but not so much in the inner part where the corals dominate. And I was wondering if you have some ideas about why that could be.
Yeah, I think it is mainly due to the geomorphology of the system, because when we arrive into the innermost part of the lagoon, corals are in an area that is, I think there are a little bit less current compared to where sponges are, because are really in the middle. So at each high tide and low tide, they are really a lot of feed. And corals also, we found them in the first pool. So where there are, we hypothesize that it could be accumulation also of heterotrophic input, and this could be one of the main facts that can lead the distribution of organisms.
Mm-hmm. Okay. Another question I had and that I found interesting reading was that some of the reference reefs that you use that are outside of the lagoon, you wrote in your thesis that because the volume of seawater discharged in 6 hours is so large, it actually also affects the area outside of the system, even if it mixes with the main lagoon's water. And you wrote that that means that the area affected by the suboptimal conditions is actually larger than previously thought. So I was wondering whether that actually means, well, whether you think that your reference reefs are actually really representative reference reefs.
Yeah. So when I talk about the more outer part of the lagoon, where I found the effect of also the chemical and physical parameter, it was just try to find something where, well, maybe yes. So here, where you see the white dots and the yellow square, I found the same fluctuating differences that I found in Boracay. But if I now answer your question, if you mean about the two control sites that I choose, I think that R1 is better than R2 as a control site, because here is seems likely also a semi-enclosed lagoon, and we have mangrove also outside here. So when we compare Boracay with these two sites, I found more differences with the external control compared to the inner between yes, the R2 control. And at the beginning we choose this control to really understand the gradient also of this environmental change in really Boracay after a semi-enclosed lagoon, but outside Boracay, and finally to the really outer reef, so R1. So I think that I could say you is that so while in Boracay we have mainly the physical parameters that were driven by the tides, in the two control sites the differences are mainly driven by the time of the day. So this is the main differences between a control and the Boracay site at the level of physical parameters.
Yeah. And what was your reason for choosing different reference reefs for the third chapter? So like the ex situ experiment, why did you collect sponges from the other side of the island rather than from these reference reefs?
Because I didn't find Rhabdastrella in these two control sites.
Okay, so not at all, not even, because I think even at the other side they're quite rare and the cover is quite long.
Yeah.
Okay. Do you have any thoughts about why they do not occur here at all?
I think that maybe at the beginning we look in the wrong site, but little by little we understand Rhabdastrella, and I think that they were more in the external reef, where there are a lot of currents and also nutrients that could help these sponges. So I don't know why we can't find these sponges in these two control sites. But yes, I think that maybe we can find a control site more near to Boracay than in the other side of the New Caledonia.
Okay, thank you. Then I have a question about the incubations that you did for the second chapter. And so again, I'm coming here from a coral perspective, but I found it quite interesting that you switched the order of the photosynthesis and respiration incubations around. And I fully understand why you did that, because the light levels were different in the morning and in the afternoon. But at least in corals, there is a difference between light-enhanced respiration and dark respiration. Do you think, is that also the case in sponges? And do you think that could have affected the measured rates for respiration?
Yes, I think that could be also the same for sponges. It's for that at the beginning we also repeated this experiment in controlled environments, because it's really difficult to perform all the incubation in Boracay at the different tide at the same moment. So yes, I think it's something that needs to be further investigated also.
And is there any evidence that light-enhanced dark respiration exists in sponges?
Sorry, I did not understand.
Is there any evidence that light-enhanced dark respiration exists in sponges? So that basically, if the sponges are exposed to light before you measure respiration, whether those rates are different than if they have been exposed to the dark before? Yeah.
It's for that we do a dark adaptation before, and we try to actually for better understand the methods that we use for the futures is we need also to better, as we said before, characterize the symbionts of my sponges. But yes, there are also for sponges a dark adaptation for the incubation.
Yeah. And then I also wanted to talk to you about your ex situ experiments. So generally, I found the experimental setup quite interesting and useful in that you mimic the fluctuation temperatures that occur inside the lagoon. But I found it a bit misleading, actually, throughout the chapter that you referred to the fluctuating temperature treatment as the 32-degree treatment, when in reality it was really a fluctuating temperature treatment. So I'm wondering where this number, the 32 degrees, comes from.
Actually, what I would like to test in ex situ is the temperature that I found in summer in 2020 in Boracay, because it's when I found the shift in sponge organic matter recycling. So I repeat exactly the same fluctuating trend that I found in situ. Actually, this in Boracay, here by here, we have an increase of also the higher peak of temperature. For example, I have here, if I found it. Yes, this one. So I investigate a little bit the temperature in Boracay, and from 2018, in the summer of 2018-2019, I have 1% of the time that organisms stay at temperature higher than 30 degrees. But we can see that here by years in the summer, there are an higher exposition of higher temperature. And so we found that this fluctuation stay like 6 degree of differences from the minimal to the maximal temperature, but like this fluctuation increase year by year.
Mm. Yeah, no, I understand all of that, but I guess my question is, if I understood correctly, the heat treatment in your ex situ experiment, it was not constant 32 degrees. It actually fluctuated from 28.5 to 33.5 every day, right?
Yeah, this is because I would like to understand if a fluctuating environment could have an effect on the incorporation of labelled coral mucus in sponges.
Yeah, so, but again, my question was kind of why, so if the temperature fluctuated between 28.5 and 33.5, why would you refer to this treatment as 32 degrees? Was that the average temperature of that treatment, or? Because
when I made the incubation, so in the Becher, I can't do the same fluctuation. So I choose the temperature where organisms were more exposed in this fluctuating temperature of Boracay. So it's for that I choose 32 as a incubation temperature.
So I guess my next question is then, this is something that we're also seeing a lot with the corals in the Kimberley, where it's a very highly fluctuating temperature environment. And what we've seen there is that these corals, and again, I'm not saying the sponges should respond like corals, but we've seen that they can cope very well with temperature fluctuations, and they can tolerate high temperatures quite well if they're only asked for a short period of time, like at low tide. But then when they get exposed to chronic elevated temperatures, like during a marine heat wave, they suddenly are more sensitive than you would think. So I'm wondering how you think about that in your system. So if you had exposed these sponges to chronic elevated temperatures rather than fluctuating temperatures, do you think that would have made a difference?
Yeah, thank you for this question, because it's really interesting to investigate this kind of responses also in sponges. So yes, I don't know actually, but what we can say to do is like simply put borax sponges at a temperature of 34 degrees for a longer period, and I'm not sure. Maybe that is dead, but suffer. So yeah, I think that is something that need to be also in this case investigated in the future, because also if we have an increase of the peak of temperature, likely this year Boracay reached also 34 degrees, but the time of exposition is really shorter, so it's like 2 hours, also less in 24 hours. So yeah, it could be really interesting to see if we expose the sponge like for months, also no more months, no, but in more longer times that 2 hours, what's happened to our sponges.
Yeah, so I guess that brings me to my last couple of questions. So how suitable do you think can these fluctuating environments really be to predict responses of the resident organisms to future ocean conditions? Because of course, on the one hand, variability will also increase, but at the same time, it's a combination of maybe increased variability and especially, for example, background warming or background acidification.
Yeah, this is the big question, the big limit of Boracay, because I think that yes, there are no perfect natural laboratories where we can study the real effect of climate change. But so the result that we obtain from Boracay need to take carefully about the global change in general. But the things that we can do is that is a real site where we can study the multiple factor of acidification, warming, and oxygenation that are the 3 main effects of the climate change. So it could be the first basis to understand how organisms could develop some mechanisms and which kind of mechanisms can develop to face to climate change. So yes, in the future, I think that there will not be the same fluctuation as in Boracay, but in average, these organisms stay at a predicted condition of future. Also, if clearly it's not the same scale of time and also in the same intensity of the fluctuation. But yes, I think is one of the best things that we have to study natural laboratory and natural variability in the natural environment of the organisms.
Okay. And then my last question, because I think I'm running out of time, is so you talked about the 3 climate change stresses: warming, acidification, and deoxygenation. And you also showed that under the various IPCC scenarios, there is about a 4% decline in oxygen content in the ocean expected or predicted. But so in that sense, isn't Boracay way more extreme? So is this really a natural laboratory for, let's say, future ocean deoxygenation, or are we actually talking about hypoxia here?
Yeah, actually, in Bourraquet, for the dissolvent oxygen, for example, we have that stay always. It was always extreme if we compare to a control site. So also when I don't know if this one. Dissolvent oxygen, yes. So, so when we look to the periods in Bourraquet, the most likely 100% of the time, the dissolvent oxygen is lower than 6.5%. That is the quantity of oxygen that we can find in a control site. So for the oxygen and also for pH, but for the oxygen in this case, we have a really extreme environmental condition, and we reach also hypoxia like 1.9 milligram of oxygen. So clearly, organisms have to be developed also some strategies to cope with this lower oxygen concentration.
Yeah, so do you think deoxygenation is a problem for these environments?
Not seems to be a problem. So maybe, yes, I don't think it's one of the main problem. The main problem for these organisms. I think that due to the fact that they are exposed to really like 100% of the time as these conditions, they develop really some mechanisms also to cope with low oxygen and also hypoxia. For example, I don't know specifically in corals, but for example, for sponges, there are some papers that say that low oxygen, so hypoxia, is not a problem for the sponge.
Yeah. Okay, well, thank you very much again. Congratulations again. And yeah, thank you.
Thank you very much.
Thank you, Verena. So, Ricardo, can you please take the floor and ask the question sent by James? I don't. Okay.
Okay, it works. Yeah, unfortunately, James could not be with us. I'm very unlucky because he's a specialist and he worked a little bit with us in Bourraquet, so the question could be very interesting to develop the discussion. Anyway, he sent me some of questions. Some of them actually were already done by some of you, so I'm just going to read a couple of us. Like, yeah, I try to make an accent, a New Zealand accent. What do you think you have learned from your work in Bourraquet? Can you tell us about other sponges in New Caledonia? And more generally, about how sponges will respond to climate change impacts. This is some impact. This is something similar to what you already answered.
Yeah. Actually, I know that there are more or less 150 species in New Caledonia, but I think that a lot of work needed to do to really identify all the sponge species. About what I learned about my study is that it really focused on a particular site, so in Bourraquet, and personally I really like this natural laboratory, but I need to perform more study on other sponge species to really understand the effect of to generalize the effect of climate change on on sponge in general.
I'm totally happy with your answer. Can you explain which climate change impact, both direct and indirect impact, are the most likely to impact sponges in the future, and why?
The most impact sponge.
You don't
in
understand
the future.
my accent.
No, yes.
Okay.
I think that yes, one that personally I found during my PhD is temperature. So this could be one of the main effect of climate change that can impact the sponge metabolism.
And what about other organisms? Because we spoke about ocean acidification, the oxygenation, etc. But what really do you think will impact coral reefs, sponges, etc. in the future?
I think that it will be the combination of all these factors. And yes, maybe for sponges we have to be a distinction between calcareous sponges and spicular sponges for the effect of ocean acidification. And ocean acidification, for example, could mainly impact corals and also the oxygenation as well.
Okay, there is a question James Bell wrote that is interesting indeed. How are environmental conditions, so we are speaking about temperature, oxygen and acidification, likely to change at your study site over the next 100 years? Will there be new extreme in these variables that sponges will need to cope with? And will they cope?
Yes, actually, as I show, I think that the main factor that could really change in the next year is the temperature, because here, for example, we just see in 3 years we have an increase of the exposition of these organisms to extreme conditions, while for actually sure for also other parameters that change, but in the last years we have always the same fluctuation, for example, for pH and dissolvent oxygen, but dissolvent oxygen could also change due to the mangrove system and also sedimentation reaction, and could be also a more hypoxia site than actually is.
Okay, I develop. We are presenting Boracay like a natural site to study the effect of climate change, extreme condition, etc. So now we have corals, sponges, etc. that resist, as you showed, about this condition. But global warming is acting now, and it will be worse in the next decade. So do you think the species you studies and the corals as well, or other species that are in Boracay now, are at the extreme level of thermal tolerance today, or do they have a other degree of compensation to stay there in the next decades phase to climate change?
Yes, I think that they need to develop more mechanism than actually we have, that actually they have in Boracay in the next years.
Okay, and last from James is that is difficult to answer. What are the wider ecosystem implications for temperature-induced shift of sponges? What
Shift
this shift you measured in detritus, etc., how could affect the whole ecosystem in Boracay, or in general, if we use this data for a general ecosystem?
Yes, if we actually we consider the Boracay sponges, so we have that at 32 degrees we have less detritus production in the environment, And for example, if we continue with the relation with corals feed in particular organic carbon also. So a decrease of this food availability could also affect feeders such as corals and detritivores. And for example, one thing for corals could be that they could have less access to heterotrophic inputs because like particular organic carbon, and then maybe they could be more sensible to resist to extreme conditions such as for increasing temperatures, such as for bleaching.
James, no more questions. Thank you very much.
Thank you.
Okay, Ricardo, thank you for James. And now, Benjamin, could you take the floor, please?
Yes, thank you very much. And also congratulations already to you, Federica. It was really a pleasure to read your thesis and also to hear your presentation today. And I must really say that similar to what the other people of the committee already mentioned, it's really impressive what you were able to do during your PhD, what kind of aspects you looked into, all the different types of analyses. And I agree with what was said before, that there is so much data there that basically all of these aspects could be ending up in, again, a new paper and a focus more in digging more into certain areas. I would now like to start with a general question about the sponge loop. And I read multiple times in chapter 2 and 3 that you were making this discrimination, what happens with the DOM that is taken up by the sponges, that it is either assimilated into biomass or it is released as detritus. Can you explain a little? So it almost appears as if these are two different directions that it goes. Can you briefly sum up what you mean with that? Also, when you look into the stable isotopes that are enriched either in the biomass, in the tissue, or in detritus. Can you please elaborate a little bit on that?
Yeah, thank you very much for your comments. So what I think is that during the normal condition, that I found that my sponge do the sponge loop pathway, and this is also interesting because normally the massive sponge showed lower detritus production, but here we enter in another big question about the methods and also the kind of technique that we use to measure detritus. But I think that when a sponge is in optimal conditions, such as during the high tide, yes, they invest more in the cellular renewal and to make the rejuvenation of the cells. While when this condition became more extreme, such as during the low tide, the sponge invests more in the biomass. Is for that, I think it is for that that I found also more carbon stored in the tissue. Also, if need to be investigated where really this carbon goes and where it's used. Because there are some recent papers also that show that when a sponge is exposed to fluctuating condition and extreme condition, they can develop some the expression, so upregulated some gene that could allow to maintain biomass and grow. So also in this case need to be investigated more, but it could be a strategy used by the sponge to cope, to have the best strategies according to the different environmental conditions.
Yeah, thank you. It now also became clear because before when I read it in the thesis, I wasn't really sure that it was that clear stated that also for the detritus production, first there has to be assimilation into cells, so basically into tissue. So even the detritus that is released was beforehand assimilated into biomass, and just part of this biomass was then released. But you made this now clearer. You also mentioned that massive sponges are supposed to either, when believing to some researchers, don't produce any detritus, or that at least they produce much less detritus. Can you maybe elaborate a little bit on different methodologies that are in general used for collecting or for measuring the detritus production, and how this might also be have an effect on the detritus rates that will be measured in these different studies, but also compared, what does it mean for the approach that you took in your chapter 2 and 3?
Yeah, actually, in the massive sponges, the article used the called the in-X method. So they use the collected seawater sample from the inhalant and exhalant pore of the sponge, and then they make differences between the water that enters and that go out from the sponges. While method that I use is the incubation method, and in this case we collected the sample. We have yes, we have the chambers, or yes, a chambers closed, and we measure at the beginning and the end of X hour of incubation the detritus or particular organic carbon produced. And there are also differences in this case to estimate the detritus production, because actually normally you can do you can subtract the live particular organic carbon to the particular organic carbon and obtain the real detritus. There are someone that also measure this directly with cytometry, but it's really difficult to really find the detritus there. And so what was the question? I think that with regard to my experiment, it's interesting because I use the one unique method, so the incubation chambers, but I found both results. So I found detritus production, but I also found no detritus production. So yeah. I think that maybe it could be better to generalize also this kind of methodology, yes. And also maybe important, it would be the time of incubation, because maybe some sponges produce detritus in 1 hour, but maybe other in more than 6 hours. So this is another point to be careful. And what next? Yes, I think for the moment it's all.
Yeah, I think that is a very good point. What you mentioned, if you do, for example, this in-X sampling, and you usually just do it over a short period of time, maximum over half an hour, that you're collecting, whereas in incubation chambers, or if you place them in a pot, or if you put the sponges upside down over a funnel, then often it's, in your case, 3 hours. Other studies also used up to 24 hours. So also the time frame that we are looking at is, of course, very different. And if we keep in mind that for, yes, Halisarca is a special case of sponge, where the sponge was initially kind of based on, but there the cell turnover was supposed to be between 5 and 6 hours. So basically, when incubations are shorter, it can very well be that if it would have been integrated over a longer period of time, more could have been collected. But of course, the advantage with the incubation is that really all detritus that is there is all captured and it all stays inside the chamber and you would be able to resolve it. Whereas with the vacuum sip, it could be when you are just, you don't know if all the particles are actually expelled from the excurrent water. And some of the detritus might also just be falling down. Talking about incubation chambers, I think it's great to use incubation chambers, but there are several people that have some reservations about the use of incubation chambers in experiments, especially when incubating active suspension feeders. Can you maybe say what could be limitations or certain things to be aware of when using incubation chambers? And are there maybe, if there are some of these, are there certain approaches how you could limit these difficulties or these problems with incubations?
Yeah, actually, I think that well, beside the first point, that is the time of incubation, I think that one problem could be at the level of the environmental condition of the seawater. If we consider the hermetic chambers, for example, so we need to be sure that the sponge is in its optimal condition to really measure the optimal the real metabolism of the sponge. Another things.
Yes.
So one aspect could, of course, also be, let's say we look at bacteria. At the beginning of your incubation, your bacterial concentration is, of course, very high. And if the sponge starts filtering the water, then by that, they always take out bacteria of the water, which means that after, let's say, 10 minutes or 15 minutes, the bacterial concentration that you have in your incubation chamber is lower than it was at the beginning. So if the sponge is filtering another 20 milliliters or whatever of water, there are less bacteria to take out. So especially when you are interested in determining fluxes of uptake, it would be advantageous to take samples, not just at the beginning and at the end. Then you would assume a kind of a linear decrease of the bacteria over time, but to rather take multiple time points at the beginning of the incubation in shorter time periods, and then logarithmically basically becoming longer. And then for bacteria, you could assume then an exponential decrease. And then you can basically, if you have the exponential decrease, you can see what at the very beginning your bacterial uptake was. So then it doesn't show you how many bacteria were taking up over time, but you kind of control for this incubation effect, that you have a depletion of your source, which is taken up. But nevertheless, that would be just something for the future if you want to continue with that, and if you're really interested in what the potential of these sponges also are in taking up. Similar, it could also be for dissolved organic matter or whatever they are taking up. I have another last question going more into the experimental design of your third chapter. You were labeling the sponges, if I saw it right, already for 4 or 8 days with the coral DOM, if I understood it correctly, permanently. And then you did your incubations either on the day number 4 or day number 8. What was your reason or the reasoning behind it that you labeled them, the sponges, already for 4 days or for 8 days in between, and did not add the label during the incubation? Can you please elaborate on that?
Yeah, Actually, the first things it was to see if we have an increase in incorporation in days, and it's for that we made incubation after 4 days and after 8 days. After looking into results, the main question that is out is if focusing on the differences in the store of carbon, labeled carbon in tissue and in detritus, and then we put beside this information to highlight the differences between the stored carbon in tissue and detritus. And for that finally I normalize the results by the days. But it could be also interesting to see this information and investigate more about these two different days of incubation.
Yeah, I think with this approach, it's definitely interesting to also see how basically you get the enrichment of carbon nitrogen in the both tissue. I think it might be a little bit difficult to compare your labeling that you're getting in your tissue, because there you basically have accumulation over 4 or sometimes 8 days. And if you compare that to the incorporation that you get in detritus, because detritus is, of course, also over the 8 days all the time produced, but you're only collecting it during a 1 hour incubation you have then in the end. And if you assume that the cell turnover for the detritus production is much faster, you, of course, have much less time for accumulating label inside that cell versus another cell that maybe has a cell turnover or a cell life of days or maybe weeks. But it was really, again, I really enjoyed reading through your thesis, and you did great work. It's also amazing to see all the ideas that you have to continue. And I think the biggest challenge for you might be now to focus and to find out in what direction you actually want to go. And I wish you all the luck for that. And I think you will do great. And I'm very happy to be part of this committee.
Thank you very much.
Thank you, Benjamin. So now it's the turn of the supervisor. So Ricardo and Yves, could you please say some words about these 3 years of thesis and what you can say about Federica, acidity, and well, just take the floor, please. Ricardo, you can start.
Yes. Wow, I'm quite tired, honestly. Of course, I have no question about the job. I'm very happy, and it has been an emotion for me this evening tonight to listen this very, very good talk. I saw a doctor, I saw a PhD. I didn't see the Italian student that in 2017 called me to make a PhD here, a master, etc. So I'm very proud about what Federica has done. Sorry. We need to consider that she didn't speak French when she arrived, and she spoke only English. Now she speaks perfectly French, and she demonstrated tonight that she can speak very well English. So I'm very, very happy about what she did. It has been a challenge, this thesis, this PhD, because it is for me the first PhD I have. I'm lucky to supervise. It has been the first PhD in Buraqué. There was another one about fishes, about oxygen effect on fishes but not not considering Buraké as a natural analog for climate change. So this PhD is for me a great result, and it was perfect because Federica did it perfectly. She published already 2 papers. One is on a pipeline. More results are quite written, ready to send. She did it in 3 years, and the talk was perfect. I'm very lucky to have Federica as a student. So I really hope that the passion she has for science will not stop in the future, and will get the possibility to make science abroad and have more experience and become a confirmed researcher. She's a doctor for me. That's all from my side. Thank you.
Thank you very much.
Thank you, Ricardo. Yves, do you want to say something?
Yes. Federica, if my English is too bad, tell me, I can switch in French. My French is much better. Okay. Thank you for your presentation. It was very clear, very didactic. I just have a few comments, no question, because we already discussed before your presentation on during all your work. So now as your thesis is finished, I only have 2 small regrets regarding the work. Very small regrets. The first one is that due to my teaching activity, I was not able to manage my time well enough to come in the field for campaign with you and Ricardo. Bien dommage. And the second regret is that I had initially believed that it would be possible to carry out some sponge grazing experiment with potential consumers, such as angelfish, nudibranch, and so on, and to see, to study how the sponge organic matter is assimilated by such consumer. And this could have been monitored, due to some biochemical markers such as stable isotopes or perhaps fatty acid, I don't know. But due to the scarcity of this consumer in the Buraki Lagoon, we were not able to do such experiments. But the question remains, and I hope we will have the opportunity to look at that question. Pardon. I would like to conclude with a few personal words, if I can. You are a person with whom it was very, very pleasant to work. You were very invested yourself in your work, but with simplicity and humility, and I like that. And it could be trivial to mention it today, but I found always important to have a little word on about the human side of a PhD student, and not only about science. And just to finish, I would like also to thank you for the time you spent with teaching ecology to our student here in Nouméa. It was another part of your work, even if it's different from your PhD. That's all. Thank you, Federica. Thanks to all.
Thank you, Will.
So it's easy to speak at the end because everything has been said, but it's less easy, of course, to speak after the expert. And I want to thank the members of this committee for the diversity of the aspects addressed and the diversity of the question which allow Federica to share mastering of the subject. And of course, I join the congratulations, and I personally I am very, very happy, Federica, to see your work achieved in time. You have done a very big job, and I am very happy to be part of this committee, even I am not an expert, as you know. But I enjoyed to read your thesis. I learned a lot about the sponges, which I think it's perhaps as neglected as the algae. So I am happy to see that people work on neglected groups. Okay, so I am quite sure that a big job we make without any doubt reference on the matter and for the site, of course. And I congratulate your supervisor to, to have designed with you this project. And I may congratulate you for your big effort to publish most of your data, and I hope you will have time to publish all the data because your thesis is very rich and with very original data. Okay, so I have no specific question because the one I had have been already asked and you gave the answer. And well, I try to have a question on the algae. If I understand well, and if I remember, because I went on Boracay site, the algae were more abundant in the inner site, right, Federica?
Yeah.
So
Sorry,
do you have any explanation for that? And do you think that they can play a role, a specific role for the sponge feeding?
Yes, of course. Sorry. Yes, we found also algae in the middle, so where we found sponges. And actually, I'm focused on the dissolved organic carbon that is released by corals, but we have to know also that sponges feed on dissolved organic matter released by algae. Then it will be also really interesting to see the proportion of these two different sources of carbon used by the sponge, and particularly also in Boracay, if this could play also differences in Boracay Reef compared to an external reef.
Okay, so thank you. So I would like also to say something about your implication in the team, because when you start your PhD, I was in charge of the redirection of the team Entropy. And well, I want to say here that it was really a pleasure to have you as a student in the team. You make very big effort to be involved on the activity of the team. And I have here to compliment you for your big effort to learn the French, because you are now very fluent in French. And I'm sure that you will be able to deliver your defense in French. So I think it is important to say that here, because it's an example of what you have done during these 3 years to be very, very well implicated in the team, and well, as I can say that, well integrate to the team. And this is, I think, a very important point to highlight here, because it's your own work, but you have done it regarding all the people and the diversity of the activities of the team. So thank you for that. It's an opportunity for me also to thank the Aquarium des Lagons, because with their support, you were able to provide the very nice results on the chapter 3 of your thesis. And for me, it was probably one of the masterpieces of your work, all this part in ex situ experience. Thierry says something about the, well, he had some regrets about the lack of experiments on the sponges from the the East Coast site. I don't remember the name of this site, but what I have to say here, because you did not say your difficulty to find the species outside of the Buraque. And I remember how it was really difficult and how you were pugnacious to find this species outside the Buraque. And I am sure that you found it on the early starting of your thesis. Probably you will make some experiments on the specimen from the control site. So I think it's important to, well, just to say here that it is not so easy to develop experiment in the field. When you expose it, it seems to be very, very easy, but in fact, it is sometimes very difficult. And I have to attest that here. So thank you again, Federica, for your pugnacity. I think that all the things have been said, so I will hand my comments here to thank again all the committee, to thank you, Federica, for this very, very big job. And I saw that you have plenty different ideas to go further. So what I can wish you is to find the right way with the right question. And I hope that you will find some granting support to offer you the possibility to continue on academic system, because I understand that this is your priority. And to finish, I have to say some thanks to the university, which hosts us every time with a very late hours, and a special thanks to the technical staff, which take care of all the defense, and it was just perfect. No trouble, so, well, magnifique. Merci beaucoup, Federica, et merci au comité.